So we need to talk a little bit about how DNA works. So we know now a DNA molecule is made of nucleotides. It's double-stranded. The DNA has a series of letters in it, bases we call them, A, T, G, and C. And the order of those letters are what determine an individual's traits, their characteristics. Those are the genes. So there are times when a cell needs to copy all of its DNA. If you remember when we talked about cell division, mitosis, a cell copies itself. In order to do that, it needs to copy all of its DNA to go into the new cell. It does that through a process called DNA replication, basically copying the DNA. And the way it works, okay, is the DNA, because it's two sides, it kind of unzips down the middle. The two halves of the DNA molecule are held together with these pretty weak bonds called hydrogen bonds that connect the two halves. And they're pretty easy to separate. So when the DNA molecule needs to make a copy, it unzips, so there's the two halves of the DNA molecule, like you see here. And then new nucleotides start to fill in these gaps. Okay? Each strand serves as what we call a template for the other strand. Okay? And the bases fill in, and when you're done with DNA replication, you have two identical copies of the DNA. Okay? It's been replicated. Here's a little picture that kind of just shows it. So you see this DNA molecule has unzipped. The gray is the original strand. And then, as it unzips, new nucleotides start to fill in. And this is eventually going to become two completely separate strands of DNA, but they're going to be identical. They'll be exact copies of each other.
Oh, yeah.
So the DNA unzips, the new bases kind of fill in. I have a game here I'll show you that shows how DNA copies itself.
Wait, can you go back?
So the DNA unzips, new nucleotides start attaching until it's completely been copied. All right, so we say that our DNA is our genes, and it is, but what it really does is DNA contains the code, the recipe for how to build different proteins in our body. So our bodies are made of mostly protein. All the enzymes in our bodies are made of protein. And that's what DNA really does. It tells how to build these different proteins. These proteins, they do most of the work in the cell. This is an example. This is a proteins. They're very large molecules, very complex. They have a specific shape that allows them to work. If you remember back to when we talked about cells, where are proteins made? What organelle in a cell makes proteins? Trying to give you a way to remember that. Ribosomes, yeah. Proteins are made in the ribosomes. And a protein is made of a whole bunch of smaller molecules called amino acids strung together. There's about 20 different amino acids in living things. And a protein's made of thousands of those amino acids put together. And again, like the DNA, what order you put those amino acids, that determines what the protein's going to be like. Okay? It determines the shape of the protein, it determines what the protein does, what its function is. So this is a key thing to know, this top sentence, that really what DNA is, those A's, T's, G's, and C's, they're telling your cells how to put together various proteins. Okay? And that process is called protein synthesis. Synthesis means making protein. So, you know, DNA has the recipe. That's what a gene is, a recipe to make a protein. Well, where do we say, where is the DNA in a cell? In a nucleus. But here's the key thing. It can't leave the nucleus. DNA has the recipe, but it's stuck inside of the nucleus. Where are the ribosomes? Are they in the nucleus?
No.
No, they're outside. So somehow in the cell, this recipe needs to get from the DNA in the nucleus to the ribosomes outside the nucleus. And the DNA can't go there. So there's a solution. What happens in a cell in protein synthesis is the gene, the section of the DNA that's the code, the recipe, it gets copied into a different type of molecule called RNA. It's called messenger RNA. We abbreviate it mRNA. Do you think this mRNA can leave the nucleus?
No.
Yeah,
Yeah, where's it going to go?
to the ribosome.
To the ribosome to give the recipe to the ribosome. And that's what happens. The instructions from the DNA are copied to RNA, messenger RNA. Then that can leave the nucleus. It goes out into the cytoplasm, attaches to a ribosome, and then the ribosome starts reading the recipe and making the protein, putting the correct amino acids in order. There's one last type of RNA that helps out. It's called transfer RNA. And its job is to bring in the correct amino acids. So remember, there's 20 different amino acids. The transfer RNA brings in the amino acids, one after the other, so that the ribosome can link them up. So let's look at a couple things here. So protein synthesis is producing, making a protein. That's what synthesis means. It means to produce. Caleb? Let's finish up this first. So this is going to repeat several times. So what molecule is this purple thing?
I don't know, Mass.
Should know.
Oh,
Double
It's a double helix. It's a DNA molecule, right? How did you know that? Caleb, how do you know that was a DNA double helix? Yeah, it was that spiral shape. Okay? So this is a DNA molecule. This has the recipe. But, as we said earlier, this recipe, it's stuck inside the nucleus. So a copy needs to be made in the messenger RNA, and that's what was happening. We'll go back to it one more time. So to use this recipe to build a protein, what happens is first the DNA unwinds, a section of RNA is made to copy the recipe, and this RNA is going to go out here to the ribosome. This is the RNA. Once it gets to the ribosome, these transfer RNAs start bringing in the correct amino acids, which get linked together. Eventually, it's going to form a protein. Now, a normal protein wouldn't just be 4 amino acids, it would be hundreds or even thousands of them. But the process is the same. DNA goes to RNA, which leaves the nucleus, goes to the ribosome, and then transfer RNAs start bringing in the correct amino acids to be linked together to actually form the protein. So this mRNA sounds like DNA, and it's very similar. Remind me, what does DNA stand for? Deoxyribonucleic acid. RNA stands for ribonucleic acid. And it's similar to DNA, but it has some differences. First off, it only has one strand, one-sided. It's not a double helix like DNA molecule, it's a single helix. It can leave the nucleus. It's a smaller molecule. So although DNA is stuck in the nucleus, messenger RNA can leave the nucleus. If you look over here, there's a difference in the bases. What's the difference? What base do you see here that you didn't see in DNA? U. And what letter has it replaced?
T.
T. So RNA doesn't have a T, it has a U instead. Every 3 messenger RNA bases is a code for one of the amino acids. So GCU is the first, we call it a codon. That's a code for a certain amino acid. ACG is the next one. It's a code for a different amino acid. So it's telling the cell which amino acids to link up. You don't really have to worry about this, but we even have a chart that we can look up these codons. So what's the first codon here?
1.
What letters is it?
GCU.
GCU. If I go to my chart, I could look up GCU. That's a code for an amino acid called alanine. If I look up a code AAG, that's a code for AAG, lysine. So every set of 3 letters is a code for a different amino acid. So it's giving the recipe, telling the cell how to make a protein. So just to summarize, protein synthesis. First, the gene is copied from DNA to messenger RNA. That messenger RNA leaves the nucleus, goes out to the ribosome. When it gets to the ribosome, every 3 messenger RNA bases is a code for 1 amino acid. The transfer RNA brings those amino acids there. The ribosome connects them to actually form the protein. And those are the steps. Eventually, the protein's finished, it's completed, and it gets released to go do whatever it has to do. Just another view of it here. What molecule is this blue thing? That's the double helix. That's DNA. It unwinds. What's being created in here?
Protein string.
Nope. This is the messenger RNA to leave the nucleus. Goes out. Here's the ribosome. This is the messenger RNA. You can see the amino acids being strung together, one after another, being brought here by the transfer RNA. Okay? So that's how proteins get made. Recipe gets copied, brought to the ribosome, so all the amino acids can get strung together. Any questions?
No.